Okay, well, welcome to the Informatics Power Talk, the weekly seminar series. We're very happy to have Dr. Marcin Joachimiak, if I pronounce it correctly, sorry. Did I pronounce it closely enough?
Yeah, close enough.
Yeah, okay. And then I think Marcin and I got to know each other from last year, but I think in our career trajectory, because of the so much stuff that he did in bioinformatics that trace back to more than 20 years ago, I'm pretty certain that we may have crossed the paths just without knowing each other by face. Marcin is a staff researcher and software developer in the Environmental Genomics and Systems Biology Division at Lawrence Berkeley National Lab, which is actually situated also on the UC Berkeley campus. He has a very diverse training that started as a mathematics major at the University of Chicago. And then he studied biophysics at the UC San Francisco. He had even just ventured into the startup world, as everyone in the Silicon Valley does, to found a company called the 5' Therapeutics. And then he came back to UC Berkeley and joined Brenner's lab. I presume it's a Sidney Brenner, the Nobel.
Stephen Brenner.
Oh, Stephen Brenner, but also is a very highly recognized person in bioinformatics who won I think a major bioinformatics Benjamin Franklin Award many years ago. And then he's currently working on many projects, and I'm sure that today's topic does not summarize all the work that he has done in the past over 2 decades. I was looking up online and was able to find this very interesting project even today, like herbicide screening for wheat, identification of novel drug targets and inhibitors for malaria, and gene finding from mouse genome, phylogenetic analysis for the global ocean survey using metagenomics, microbial functional genome analysis pipeline. His work involved many practical bioinformatics skills that range from data modeling, data science, machine learning, ontology, knowledge graph, visualization. If you can think of anything in bioinformatics, and he may have already done it. So it's going to be a very interesting talk, and let's welcome Marcin.
Thanks for the introduction, Jake. That's very nice. I guess it's true that I've worked in lots of different projects over the years, but maybe that's just the nature of bioinformatics, right? As you move in your career, get different funding, you're part of different projects, you get to sample different parts of the bioinformatics world. So thanks for the intro. It's a great pleasure to be here. So today I wanted to tell you about how to teach a computer to learn about microbes on this example of microbial knowledge graph learning. Okay, so last 2 years of the pandemic, I have rewatched the entire Star Wars saga with my son. And so this is maybe just a facetious kind of intro to say that, maybe in the near future, teaching computers will look very different than we think about it today. Of course, we're really just starting in that whole process. But you can imagine some very fast interfaces for complex data that make our life easier. Of course, we're not there yet. Okay, so I wanted to back up and kind of go back a little bit in time to some history of science. And so this is back to the 19th century, where people are trying to understand and really form theories about human evolution. And there was this idea about the missing link. So people were going into the field and trying to find fossils that would help explain the evolution of humans, essentially. And so there's some fairly interesting, and from today's perspective, maybe funny views about this. So this is one from a really German advocate of Darwinism. But this is actually a painting by an artist called Benjamin Phillips. Like, it's called "Evolution Gets Us Nowhere," and I like this just as an example, an extreme example of taking data and attempting to structure it. So, depending on the person and their experience, their worldviews, right, you may get different solutions or proposals for a theory, especially when you have incomplete data and there's still not consensus in the field about what things mean, right? Okay, well, so I work in microbes, at least in the last 15 years or so. Before I did a few human-related projects. Now I'm starting to get back into human as well. But there are certain areas of science that are more developed, that have a longer history, and maybe the data they work with is more accessible. So for human evolution, it was a lot about anatomy, comparing microscopic elements of our bone anatomy, for example, to try to interpret how evolution and species changed over time. And so here, if you have a pretty good context of time and geography, you can place your findings in that context. And now you can have a portion of information that's reliable. So you're fairly confident that these time ranges are correct. You, of course, know the locations. And then on top of these, you can posit some potentially other information that you may not be confident about, but over time will get filled in. And so these are these question marks. So I think if you think for microbes, we don't really have a very structured view yet, especially in terms of distribution of functions and distribution of taxa and how they coexist and interact. And so we have a lot of question marks and very little evidence, hard evidence to hang things on. So hopefully this is kind of an intro for you to get a different perspective on how we can structure data into knowledge. Okay, so just a quick overview. I'm going to talk about knowledge graphs, introduce some concepts, talk about the design principles we like to use, and also our KG construction framework. KG will be just shorthand for knowledge graphs. And then we'll talk about KG microbe, the motivation for that project, how the BioLink model fits there in terms of standardization, more design and construction, and some graph learning applications. And there's an analogy here, at least from the pandemic, where scientific projects have some analogies to cooking and baking. So here's just a focaccia that happened recently from raw ingredients, and I think this is actually a pertinent analogy where you start with raw data and make some knowledge product in the end. Okay, well, this is maybe a messy slide. It is really just to show one interpretation and view of how we learn currently. Things have changed in the last decades, mostly because we can publish lots of different pieces of scientific research, not just papers anymore. So that includes data and code and maybe intermediate findings. You can do preprints. And so people have a different perspective now on how they can be inspired to design an experiment or start a new project or maybe change the direction of what they're doing. So there are a few elements here, and I think people maybe use them in different ways. But I think everyone will agree that we're in a rapidly changing time for how research is done. Okay, so we do need some starting point. And we argue that graphs are a powerful data structure, but both for machines and humans, actually, and especially so for biological data. The reason is that biological data is fairly sparse. You can have different types of measurements for different samples or different related information. So you can't really quite align them. But in a graph, you can store all of that information and represent the links as you see them. And graphs can scale to many dimensions. There's some nice hardware advances recently that improve storage and can compute. And there's a very rich set of querying languages that you can perform complex search on data that's been graphified. So if you're not familiar, on the right are a few properties of graphs. They have nodes and edges. These can be homogeneous or heterogeneous. So homogeneous would be of the same type. For example, a protein interaction network would consist of just protein nodes, whereas something like the Monarch Initiative would have, highly heterogeneous mix of types. And same for the edges in a graph. Edges can have weights, they can have directions, you can have multiple edges between nodes, and then there are properties that you can embed into the graph as additional information. And I think something that's underappreciated is that mathematically graphs and matrices can be analogous. You can transform one into the other. Of course, you may run into the sparsity issues that I mentioned. But the important piece here is that we have a much longer history of methods developed for matrices. So there is likely some cross-pollination that can happen here, and maybe some additional collaborations to happen, maybe even for combined matrix graph methods. So I think that will be an area that will continue to develop. Okay, so what is a knowledge graph? Here is just an easy example, kind of accessible example based on actors, the characters they play, movies, and the genre. And so here you can quickly see that these two actors share a genre, science fiction. So you can imagine how this can work in the same way for our biological data or biomedical data. But of course, you need some standardization for how you label the actual nodes here, their categories, and their relations. So I'll have much more on that coming up. I mentioned the node properties, so that essentially you can think of that as containers for additional information. Things like provenance or metadata for a node or edge can be stored there. Knowledge graphs also rely on standardized identifiers, so both for the entities, the relations, and the properties ideally. And they're also tied to the semantic web and ontology worlds. And so they can make use of synonyms, alternate IDs, mappings to harmonize the data. And you really need this to perform normalization of the entities where you merge things that maybe have a different ID, but refer to the same concept. So that's actually a big area, body of work that's still under progress today. Okay, so moving on to some design and organizing principles. This is all really spearheaded by Chris Mungall and his group. So it's one group that I work with. They're really kind of excellent in thinking about this, and they're very practical. So the decisions are really driven by operationally what will work the easiest and the best way for the most people. And so one outcome of that is that the graphs are stored in this human readable tab separated format that has a few required columns. They're showed here for nodes and for edges. So it's actually a fairly light format with some requirements that the ID for nodes is a query. So it has an identifier and then a standardized prefix. And so you can, you would essentially store all of your entity and edge relation information in this way. And you can also add additional properties, additional columns, and that's up to the user to implement. We can easily load these files into different graph stores, and so that can serve queries and web APIs, etc. And I'll have a lot more about the BioLink model, but the categories and the relations here are sourced from the BioLink model, so you have a standardized kind of vocabulary to refer to how you model the knowledge here. Okay, so a few more organizing principles. I mentioned the normalization, so that's a key aspect where, either through mappings or some other process, you're able to collapse IDs that refer to the same entity. This is a big problem for genes, for example. There are some gray areas that maybe require special decisions, whether alternative transcripts are the same concept or need to be separated. So that really will be determined by the purpose of your knowledge graph and the tasks you want to perform on it. The BioLink model also provides lists of valid prefixes, and you can even turn these into inequalities. So this will allow you to have a preferred namespace for your entities and relations in the graph, but it also allows you to have mixed namespaces if, for example, a PubChem chemical is not found in the Kebi ontology, just as an example. Another aspect is that you want to target subsets of the data to satisfy your KG purpose and the tasks. And so say you're working on a drug repurposing project, you might not want to include chemicals that are metabolites, just drugs, right? And so those are decisions also that have to be made. And then just as a As a guideline, we don't ingest data from secondary sources. So, for example, no protein interactions from a chemical resource. Okay, and then all of this is BioLink model compliant, and I'll tell you a bit about the BioLink model shortly. Okay, so supporting all this is a growing toolbox, and these are just a few examples of the pieces that underlie these projects. So we have a template in GitHub that you can fork to create a new knowledge graph, and KG Micro is currently the parent of that, simply because we'd added some new technology there based on named entity recognition and also some ontology tools. We also use this KGX package Python library for really knowledge graph exchange. It also performs merging operations and gives us summaries to track progress of the individual steps and then the final knowledge graph product. I mentioned the NER methods. It's part of the Monarch initiative that we're part of. This is essentially a wrapper API to different NER methods, Ogre++ and Spacey currently. And I'll have a little bit more on how that works coming up. We also have some tools that operate directly on ontology files. Robot is one of these. Mostly we use this to prune ontologies or perform some kind of search. And then finally, maybe the most exciting thing here has been a very fruitful collaboration in the last 2 years, actually just during the pandemic, with the Monarch Initiative and an Italian group of deep learners. We've developed some new graph processing and learning methods, and I'll show you some results of that later. Okay, so to kind of tie all this together, we have this KG Hub concept, so Knowledge Graph Hub. It's a reusable platform for standardized construction of knowledge graphs. And it comes with a few guidelines that, again, I think are just driven by practicality. So the knowledge graph build should be in a GitHub repo. It should contain reproducible ETL code, so extract, transform, load. It should make attempts align with the BioLink model or otherwise standardize. Obo Foundry ontologies are preferred, and that's simply because they go through additional validation and also have guaranteed file formats available. So it just makes the process easier. Of course, if your ontology is not found in Obo Foundry, then you have no choice. You're responsible for the data sources and evidence, so there's no additional validation that KG Hub does at the moment. It's kind of trusted by as on face value. Again, the downloads and versioning are required, including in this simple interchange format, and then some license and guideline requirements to ensure that these are all open for community contribution. So that's kind of the underlying philosophy here, and that really comes from Chris Mungle and the Monarch Initiative, and I think the ontology world, where there's just a lot of collaboration and kind of group decision making. So I think that's the nice aspect. So in the bottom here, I'm just showing the main steps in the KG Hub construction process. And this is really how you initiate a build. It has these specific directories and steps to perform. So there's a download data step. There's a transform data step that really usually is the most work, because that's where all the decision making and creation of knowledge from data happens. And then there's a merging step that can in theory create multiple knowledge graphs in one operation. And then of course you have some downstream applications of all this. Okay, so we have a few different knowledge graphs in the KG Hub currently. We're starting to get some economy of scale where we can begin to reuse both parts of the template, some source data that has been transformed, and actually even entire knowledge graphs. And so KG COVID-19 was the first here. That was a really great project with Justin Rees. And then it has kind of mushroomed since then. I should also point out to you that KG Obo is a knowledge graph based on all the Obo ontologies. And so that really is a very reusable object that has both the TSV files and the transforms for the ontology in Obo. And so you can imagine that as additional uses beyond just knowledge graphs, and you can use those files easily for enrichment analysis, et cetera. So there may be other aspects here beyond the knowledge graph world. And then, as I mentioned, KG Hub is integrated with these different tools, including a set of algorithms for graph learning. Okay, so I'm going to move on now to microbial world and KG microbe. We're probably all familiar, but we humans are a tiny little branch on this green subtree on the right. Can't really see us. And then the vast diversity of life on Earth are microbes. Bacteria, archaea, and viruses are not even represented here. So KG microbe focuses on prokaryotes at the moment. But to kind of set up the context more, I think they're, compared to biomedicine, at least, in the kind of the human biology world, microbial research has some unique challenges, I think. One is that there's just a huge number of taxa and strains and variants, of course. And then what follows from that is that they have a large repertoire of functions that are deployed in a large number of environments. So there's a lot of combinatorics happening here. And of course, we haven't sampled enough to have a very clear picture in a lot of cases, although maybe for human, that is actually approaching something where we're getting a more global understanding of microbiome to human interactions. There's this finding that a few kilograms of our body weight are microbial cells. And then because of this large repertoire of microbial functions, maybe these different interactions of health and disease are now less surprising, but some are actually quite amazing. A lot of amazing literature about effects or correlations of microbes with different aspects of human health. And finally, just that microbes are a large fraction of biomass on Earth, especially compared to animals and even humans. So, so hopefully this convinces you that they're important. Maybe we already all agree on that. So that's kind of the complexity in the wild of microbes. There's also the complexity that humans have created, not on purpose. Part of this is that microbial research actually has a fairly long history. And there have been many different efforts over time that were underfunded, were lacking long-term funding. And so, for example, we have 4 kind of competing taxonomies that overlap to a large degree. They have some differences in how they model things, but the ones that are based on sequence data tend to be leading the way in terms of inclusion of new taxa. And so there's some important differences there that will require some merging or other solution. I think it's similar for functions, functional classification and traits. We just have multiplicity here. All of these projects have value. They probably have some non-overlapping information. So probably in the future, there might need to be some unification that has to happen. So not of any, this is no one's specific fault, but I think underfunding and kind of long-term visions were lacking here, especially compared to human research. Okay, so now moving on to KG microbe. This is our kind of parent reference microbial knowledge graph. One maybe implicit concept in the way we build these KGs is that we pick a few reference ontologies that we expect to cover the terms, entities that we will find and see in the relevant data for that KG. And so in this case, I'm showing you the example of this trait table that was published by Marcin et al. They compiled literature and textbook information about 12,000 taxa. And so using NER methods and these ontologies, we're able to link the information in that large table across the ontologies. And a KG microbe is actually bigger than this. We have a few more data sources in progress and a large list of tickets on the GitHub repo. And I urge people to look there if you're interested. And we're also open to suggestions about data to include. Okay, so underlying all of this, just more generally, are these data harmonization challenges. They're kind of akin to the microbial world that I just showed, where people model things slightly differently, or maybe very differently. And so you could try to align these different data modeling schemas, but usually that won't work so well. And so you're kind of left with picking the pieces that are usable in the end. So this is really a problem of resource interoperability. We lack standardization, and we lack mappings between different resources. So to try to address this, the translator project funded by NCATS, biomedical data translator, was set up to provide APIs to different data resources for human research, essentially. And then these APIs are designed to feed into a reasoning agent that can then take as input a free text question, even from a patient or a doctor, researchers, and then answer that question based on these data inputs. And so, that's a very grand vision. It's not going to happen, I think, very quickly. But the big problem to solve first was this data standardization to be able to link all these different data resources together. And so the idea was this high-level data model to represent knowledge. It's designed really to bridge different vocabularies and ontologies. It's agnostic to the type of graph you work with. And really, it's not restricted to graphs. It's just the graphs are important for a translator. You could just as well use this model to annotate statements or data in tables, other structures like JSON, for example. And at its core, it consists of entities, associations, predicates, and properties. So I'll kind of quickly now go through those different aspects. It is designed hierarchically, so every entity or association in the biomedical model has a parent. So here's just an example of a gene to disease association and its parents. And you can have different flavors of these associations. The predicate is really the category for the relation. So in this case, it's implicated in, but the exact association is much more specific. It's gene to disease or phenotypic feature association. Okay, so entities are, you can think of as nodes in a graph. They are arranged in a hierarchy. Their root is named thing. And there's actually a very nice website to browse these hierarchies with a search interface. So even though you kind of have to learn some aspects of this model, I think because of that web UI, it's actually fairly quick and not that bad. Okay, and then each entity is a class. It contains mappings to other ontologies. And then I mentioned the identifier prefixes. So this is an example of valid prefixes for a gene class. And of course, this is a community effort with a GitHub repo. So people make tickets and make requests. And this is a growing, living project. Some of these higher level terms can be used to categorize entities, which can be useful for things like enrichment analysis or grouping or some interpretation. So that's actually, for me, is one kind of primary utility here, beyond just the data standardization, is that you gain kind of a higher level understanding of how different entities are related, maybe genes from different species, right, or et cetera. Okay, and for associations, these would correspond to edges in a graph. Again, it's a hierarchy. The root of that hierarchy is association. And these actually get to be quite specific. So I think this is where the bulk of the growth in the model is these days. It's currently covering a lot of human health biomedical applications, and it's quickly growing on the environmental and kind of more microbial side. So I think this will be kind of generally useful in different applications. Okay, so associations connect in two different nodes, a subject and object. And you can define the association, you can define additional properties. Some of these will be important for provenance or as metadata. So for example, provided by would give you the source of that edge or node. Similarly for evidence, if you're making an assertion that a gene is causative for a disease, for example, you can also include DOIs for publications. And then importantly, certain associations can have additional properties that are specific. And so for example, in a disease to phenotype association, you could have a frequency qualifier. And so this allows you to attach the expected data to this kind of semantic knowledge. Okay, and predicates are just high-level relationships that you can think of these as categories for the edges. So similarly to categories for nodes, you can use this again for enrichment analysis and interpretation. Most of these are sourced from just the standard relations ontology. Okay, so just to summarize, it's a common dialect for representing knowledge. It has a community of designers and users. Anyone is welcome to join. Bridges different types of graphs. There's actually growing now code for mapping existing models to BioLink. So I think that will just make this very painless. And importantly, it's this shared space for discussion on these important concepts for knowledge modeling. Okay, so that was the BioLink model. Now I'm going to move to kind of one application here that's tied to microbial data. So it's another project we have around the NCBI biosample collection. So it's metadata about samples. There are 28 million samples. About half of those are human-based. So there's a lot of information about human disease and health there. And then the rest are mostly microbes with some eukaryotic data. So this is just a kind of toy sample metadata record. The field names are in italic and the possible values are listed there. So you can see there can be a lot of variation here. Some of the things can be difficult to reconcile. So you might end up using different portions of the data in different ways. For example, short versus long reads, or environmental terms sourced from different vocabularies. But what we're most excited about are these sample measurements, specifically for the context of microbes and their environment. And there's this very rich data. It's unfortunately fairly sparse, and it has a lot of repair issues where people use different units, don't provide the unit, or have some other kind of variation relative to what other people have entered. So often these are sample records that have been uploaded to a resource like NCBI. So to deal with this situation, we have a project to build an API to repair and standardize this metadata. And it's really targeting these 4 different areas. So repair of location values and lookup of that location, repairing measurements. This is probably the hardest problem, simply because for some cases there are lots of units, or maybe you're missing a unit, you might have to infer that. And so even though we can use some existing packages that are fairly good at parsing units and values, I think that will probably take the longest time. And then we also are able to repair text using these NER methods, so named entity recognition. We provide a dictionary of standardized terms, and then again, standardizing with ontologies. So this is just the command line example. And so an input record for a sample metadata to be repaired may look like this. One thing that you get that's really nice is there's a report of all the different cases that are identified and which ones could be repaired. We can also use this to kind of track progress of the code as the different methods gain better coverage of all the cases. And then it also outputs this cleaned repaired record, where one aspect here is splitting units and values, maybe listing the full name of the value, et cetera. So lots of different things can happen here. So I mentioned there are 28 million NCBI biosample records. It's actually a huge collection. And we think this will be a really important resource to enrich machine learning and other analysis, simply because you can pull in additional data for your sample that you may not have any access to at the moment. Okay, so back to KG microbe, just a few construction steps in a bit more detail to give you a better idea of how this works. So I mentioned the Madden et al. Trait data table contains information on the level of taxa. So it's kind of derived across samples, if you wish. And then these are the columns that we identified as useful and amenable to this NER step. So this is their list. And then the next step, we transform them into subject, object, predicate data, so triples. Each row in this table corresponds to an organism, and it has these properties. Not all of them will be present. And so the first step is to identify which ontology can support which column. So for chemicals, we use Kebi ontology, environment terms Envo, functions and pathways Go, and NCBI taxon for taxa. And then the next step is to define what types of triples each of these fields will produce. And so here you can see that all of these are organisms to some feature. And then finally merge all of these pieces together into one nodes and edges file that will represent your knowledge graph. This is the NER step, where you take sorts ontologies, transform them into a format that's useful here, and then plug them into these NER wrappers together with the source column that you want to standardize. And the output that you get is a new column with standardized values based on the ontology that you input. And so usually, have a little bit of loss of information, depending on the ontology coverage. You could maybe combine multiple ontologies if necessary, but in the end, you get something that's comparable across the rows of this table, and then also aligned with additional data in your knowledge graph. Okay, and then I also mentioned we use some ontology tools. This came about because the NCBI taxonomy is quite large. It's over 1 million nodes and growing. And it has presented some issues for us, specifically for graph learning, because this tree is connected at the very top with a root. And we didn't want this kind of trivial link between everything through the root of life. So the top of this tree was trimmed using this robot tool. There are also some kind of oddities like 40,000 Arabidopsis strains, also some branches of the tree of life that are maybe less relevant to microbes. So these were again trimmed out, and we use the robot tool and its commands to kind of process the ontology and take subsets. So that's, again, I mentioned that was a new piece added with KG Microbe and now part of the KG Hub template. So you can apply this to, maybe chemicals or environments, other large hierarchies where you might not need all the pieces for your purpose. As part of the KG Hub, we also get this dashboard. So this is a kind of both summary and breakdown of the KG contents. And we use this as kind of a light QC element because the KGs are built automatically. So we'll identify a new data source, write the code for the transform, and then using a automated build process, kind of push the button and get the built knowledge graph along with these other diagnostics. And so here you can see the breakdown for the categories for the nodes and edges. And this may be also useful to fill in certain entities that you think are underrepresented. In this case, for example, we have a large number of chemicals, large number of taxa, but maybe fewer activity and behavior. That's also because there are fewer sources for that data for microbes. We also have a different view of this down here in the bottom left. Apologize that it's too small, but this is a Sankey plot that will show the sources of your KG on one side, what their relation in the middle, and then what they map to in the biolink model. So this really shows you a kind of deeper view into the data transform and how the raw data gets transformed into the KG world. Okay, so that concludes kind of the KG construction section and KG microbe, and I'll move on to the graph learning portion. So we've been fortunate enough to have this great collaboration driven by the Monarch Initiative to develop some new graph learning methods using this Embiggen package on GPU. That's also powered by another package for graph processing. It's really quite fast. So we think it's the fastest graph processing available. It's written in Rust. And what I'm showing you here are two-dimensional reductions of the embeddings for the string protein interaction database. Each row in this view is a different algorithm, deep learning algorithm, to create the embeddings, and then the columns are different reduction parameters. And so it's really just kind of giving a flavor to show that this is over about 20,000 proteins in the human proteome. Lots of this is a fairly dense hairball if you look at just the network itself, but you can extract some other information here in relationships via the embeddings. This deep learning on large graphs is fairly recent. So the first method came out from the Leskovich group at Stanford in 2016. That was designed for CPUs with large memory. So that was about 1 million nodes. A few years later, Facebook released this custom distributed architecture that can work on the order of 1 billion nodes, but this requires a fancy cluster and personnel to manage it. So it's really not feasible for academics. And so hopefully with Embiggen, we can kind of democratize this more. It can routinely work with graphs over 100 million nodes, and it's actually approaching this billion limit today. So we think that will open up some new avenues. All right, so I wanted to spend a little bit of time on one of the kind of main algorithms in Embiggen. And so I'll start with the word-to-vec algorithm, which is the kind of inspiration for the graph, one of these important graph algorithms. This is called the skipgram model. So it's where you want to, given a center word in text, you want to predict the context words or outside words. So the center word here is brown. You have a window parameter. You have some neighboring words around it. And now you have a vector and matrix representations of this word data. you have a vector of size v, which is your vocabulary. So in this case, it just gets a 1 in the brown position. Next, you have a matrix, which is it has rows for the vocabulary size and then columns for the number of embedding features. The embedding dimensions, d, that will be something like 100 or some power of 2. And there can be a hypertuned parameter that you may have to find. And then you have a word representation of the word brown. And now you go through your standard deep learning gradient optimization with checking of the correct answer. And if you are correct, then you kind of keep those weights. If you're not, then you propagate your weights back and then redo the optimization. And so with this process, you're able to get a representation of words that actually has some striking properties. So this is kind of the claim. And I think this analogy reasoning has been applied now in a few different places. But this is a view of words placed in this embedding space where you can see analogies by similar angles, so parallel vectors effectively. And you can see relationships like queen is to woman as king is to man, for example, or more complex ones with a split relation, something with the gradient, right? Slowest, slower, slowest, that now has multiple parallel vectors as a structure. And so we can cast this into any data that we like, right? So in our case, it could be something like links between environments and species. So finding new species for an existing environment. Okay, so word2vec inspired the node2vec algorithm. I think the main takeaway here is that it's actually very analogous in that you create text by performing random walks on the graph. So word2vec could operate on any text, Twitter feed or research article, etc., just sentences of a document. And in the node2vec world, you create the document by performing a random walk on the graph. And there are really two key parameters. One determines the breadth first search. So let's say U is your starting point. The red arrows are moving close in your close neighborhood, kind of sampling the close neighborhood. And the Q parameter determines the depth first search. So how far you would move away from your starting point. And so these are two different ways to sample the graph structure. And there's some trade-offs here, of course. Okay, so I showed you this view and embedding space where similar angles imply similar semantic meaning. And so this transfers also to the node2vec graph world. And now you can pose this as a prediction problem. So you can say, given species X, its relation to environment 1, what are environments for species Y that have a similar angle? So that can be just through similarity. And you can make this more complex by building classifiers using the embeddings. All right, so maybe to give you an overview of where we are at this point, we started with kind of microbial source data, identifying the important sources that could support these tasks, knowledge graph construction steps. Of course, lots of things going on here. At this point, you get a built knowledge graph. And now you can run these graph learning algorithms to generate embeddings from your graph input. And so this essentially will give you a new matrix where each row is a node in the graph, and then the columns are the embedding features determined by your embedding dimension parameter. And now using those embeddings, you can train classifiers to perform predictive tasks like link prediction. Okay, so one of the ways we diagnose and kind of keep track of the embedding process is via these two-dimensional reductions. These are not only qualitative views in that you can't compare them from one to the other. It just gives you a relative view of all your data and how it relates via embedding similarity. And so these nodes are on the left, edges are on the right. So each data point is a single node or a single edge in the graph. And there are hundreds of thousands of these, I think over 1 million edges. And here they're colored by their biolink category. So I mentioned, the categories is kind of one useful interpretative aspect. And so here in orange, the Nike swoosh is most of our taxonomy nodes. There are a few other pieces sprinkled throughout. And a subset of these taxonomy nodes have nearby ontology classes. And so these are standardized terms annotated to these taxa in our graph. And we still have work to do. A lot of our nodes have an unknown category, either because the biolink model doesn't have a direct there's a ticket still that has to happen, or we have not gone through that yet. The edges actually are somewhat easier to classify. There's maybe fewer. That space ends up being somewhat smaller. And so we can use this to diagnose the KG construction process over time as we're adding new data sources, or maybe fixing a bug, or using a different mapping to categorize or standardize. Okay, so we thought about what would be kind of a valuable demonstration of link prediction here. And one of the kind of expensive things to do is to characterize, visually what a microbe looks like. Early on, there was lots of work on this because that was really the only tool that researchers had. So going back, to the 16th, 17th century. So a lot of the taxonomy names, scientific names, actually correspond to the shape of the microbe. And so here's just a Wikipedia image giving you a sampling of, quite diverse microbe shapes and organizations. And the Madden et al. Trade table had annotations of shape for a portion of the 12,000 taxa. This is the link prediction pipeline that we've implemented. And so kind of to lead you through this, you select the link prediction target, which would be some pair of entities in your graph, in this case, microbe to shape. You will go into your graph and find all the cases where a microbe is annotated with a shape. These will be your positive training examples. And then you need to define a negative training set. And this gets interesting or complicated potentially. You could go for random negative examples. However, because the data is incomplete, some of these will be false. So you'll have some pollution here. But depending on the data, you can pick some other strategies that should perform better. So for microbe to shape, we expect mostly these to be one-to-one. So one microbe can only have one shape. There are some small discrepancies there, but really we don't have that data where a microbe can go through different stages, for example, and it's quite more rare. And so you can use these to mismatch now a microbe with another shape and use that as a negative training example. So there's some strategies here that may be dependent on the exact data you're looking at. And so now with your positive negative training examples, you split them on the way you like for model training. But there's one more important step here, that you want to recompute your graph embeddings on this 80% graph, where you've withheld 20% of the edges to evaluate the model. So you've essentially reduced your graph by 20%. You compute the embeddings for the remaining 80%, you use that to train your model, and now you can evaluate the 20% of edges that you withheld to assess the performance of your model, right? And once you achieve some performance that's satisfactory, you can apply that to predict new links. Okay, so this is a result for our microbial shape prediction. We use the CatBoost gradient boosted decision tree method here. It's actually a very nice package, very fast, and performs just as well as random forest. So it's quite powerful in our hands, I think. And so what was surprising was, the fairly nice predictive performance here, even in some cases of low support where we have few examples. The recall in some cases is not as good, but the precision is surprising. And so we're excited to see this result. To be fair, it's likely driven by taxonomy to some degree. So we're testing the contribution of the different data sources here. And the reason is that taxonomy links all the taxa together. And in your graph walk, you can always visit a neighbor that may have some annotation. And so depending on that distance, that would lead to these embeddings having meaningful information for things like shape. But the graph contains other entities of interest, pathways, metabolism modes, grit substrates, and all of these can be cast in the same type of link prediction problem. And the colored bars represent the feature importance for the different shapes. And the features here are embeddings from embeddings, so they're on face value unexplainable. Of course, you can do some work to explain them. And in the top right is a connection to the matrix world. This is a histogram of the frequency of each feature across the 12,000 taxa. So you can see that most features are fairly sparse, only a few cover many taxa. And so, this is to show that probably standard matrix methods could have a lot of issues and problems here for a similar prediction problem. Okay, so I'm almost done here. I also see some things in the chat, so maybe I should, okay, I can look at those later. So just for future, we are working to combine literature data here, as this is part of the Embiggen package, so more on that in the next slide. I think there's lots of knowledge modeling remaining, and that will probably continue as our data types become more complex As our causal models, become fuller and more complete, there will be more modeling remaining. There's more custom KGs in the works around microbes, a few collaborations at LBL, and of course, we're open to further collaborations, and anyone these are all available to use or collaborate on. So either you can start your own project or work with us or just use one of these existing resources. And then also some new predictive applications. One important one is for growth conditions. It's difficult to culture microbes usually, and so that field has not moved in the 15 years that I've been at LBL. I think the predictive performance for growth is still fairly low. And also things like interventions for the environment, biomanufacturing and biomedical. And there we can think of a KG microbe as a parent knowledge graph, and these would be kind of child knowledge graphs with additional data sources for those purposes. Okay, so I'm almost done here. These are embedding views based on just natural language models using only the label and the description in either the nodes or the edges. So that goes back to that TSV file I showed you. In my mind, those labels and descriptions are actually fairly poor information. So it's shocking in a way to see any structure here. And of course, we have control over those descriptions. We can source them from other richer sources. And so I think this will be an important aspect. And this just gives you another way to create embeddings for the nodes independent of the graph structure, right? And so I think that would be an important aspect. Okay, and then finally, maybe tying into the data modeling, we're very keen on different aspects of biomes. And so there are two maybe main pieces here. There's the data on the actual environment in the wild that we sample. And then these samples are analyzed in the lab to produce different readouts, other characteristics of that environment, including chemicals and isolated organisms. And then of course, for metagenomics, we have a lot of derived analysis and summaries that can be useful, all the way down to taxa and sets of functions that are definitive of environments or that never are expected to occur there. And once you build up a compendium, so a large collection of, say, abundance values, you can also find other properties and features from that collection and then apply them back to individual samples. So I think there's going to be a lot of movement here in this field. Okay, so just to conclude, building knowledge graphs still involves some work, but the tooling is improving rapidly. And I think one proof there is we're seeing more and more reuse. The community efforts like the BioLink model, I think will enable flexible and persistent modeling. There may be other efforts like that, but I think it's just a nice example to consider. We are now seeing these interesting graph learning applications that are now possible on large data and complex data. This is just a convergence, and we're kind of at the right time. And then, of course, validation is key and still the most difficult thing, but then maybe there's some opportunities by using literature, so confirming results in new literature that you have not included in your training, and similar for data sources. Okay, so just to acknowledge a lot of people involved in these efforts. I work with these two groups at LBL, Adam Marcin with KBase, and then Chris Mungle, the translator, and other projects. It's really a great team of people, very exciting, and I think we'll have more interesting things to say in the near future. Thank you.
All right, we have some questions in the chat box.
Yeah, okay.
I guess the first question was the name of the website. It's Bill Bard, your
right,
colleague.
Okay.
Yeah,
thank you. Thank you all. Right. Got it. I think there actually may
Thank
be
you, Marcin.
There might be a few websites. I think there's the GitHub repo for kind of developers, or if you're interested in looking at the files. There is this link that Bill provided for the documentation, and that I think includes the search and the hierarchy. So probably for most people, that is the place to go. Yeah, that link, may be another one, sorry. Yes, that's right. There are teams at UAB, of course, Matt Knight and Will, that they're excellent, and we love working with them. Okay, can you comment on how we can use associations between genes, diseases, phenotypes, pathways for precision medicine? Okay, interesting. Well, so I guess there you'd have to consider what type of information you would need to be able to make medicine precise, right? And so, I guess one approach would be to do it at the level of patient data. And maybe that would have scaling problems or privacy issues. So the other way would be to maybe make some abstraction of groups of patients, right? Of groups of disease phenotypes, I guess based on variants. So I know there are some efforts out there funded by NIH and NCATS that are trying to build these resources around genetic variants. And so there is information about specific links between variants to disease or phenotypes. Of course, for more complex diseases, this gets hairier and more difficult, and so maybe there's some bleeding edge here. But I think for rare diseases, right, maybe that's one. There are some areas that are more tractable with the information that we have, where specific variants are determined to be causal, and there you can have that confidence, I guess. Does that help answer your question?
Somewhat. I actually wanted to know, can we track transition? Like in a cancer, there are several stages. So is there a way to track that?
I see. Well, so I'm not an expert. I've worked on cancer a little bit. I'm not an expert on cancer. I do appreciate that there are stages. So I guess I would say that if you have the data on stages and maybe markers, right? And it sounds like your question is more maybe about detecting the stage and tuning treatments based on the patient state, right?
Yes.
Yeah. So in that case, you would have to somehow capture the knowledge about the states and the markers that you expect to see, right? And maybe you could also have results and outcomes, of treating different states with different interventions. And so I think you would have to build up that information, assume those atomic resources exist, so variant to disease, variant to phenotype, maybe, I don't know if stage, I guess, would enter in a different way, maybe stage to biomarkers through gene expression, right, or some other blood metabolite analysis. I think that's maybe one way to think about that.
Okay, Marcin, I'm very interested in your topic. I especially like the fact that you're not trivially using the knowledge graph as collection of statements of fact, but then use deep learning to filter out the noise and then use this database for its strengths. But I also would like your comment on how expressive, a number of questions. One is how expressive is this BioLink model? Because we know that the English language is more expressive, have lots of the when, where, and the frequency and the trust embedded in the knowledge, which seem to be very essential when biologists making conclusions. But many of the knowledge representation in the past, like the bio to RDF, it just stated as a fact. And I don't think biologists would agree that four-legged animals are mammals, but then they would agree that tamoxifen is curing breast cancer, which is not true because it's only used to treat the estrogen receptor positive breast cancer, and it's only responsive for part of the cancer patient. So how expressive is BioLink in figuring out all of the nuances?
That's a great question, right? So that really is the problem. I guess I would maybe start with natural language, how we use language in literature, right? And just from this perspective of trying to process how people use terms, right? We see a lot of variation, with capitalization or misspelling or different hyphens, right? And so even just you can use natural language, making sure that people are talking about the same thing can be non-trivial, right? So even though it is extremely expressive, right? Like infinitely expressive, it's maybe too expressive, right? It's hard to compile that information together. So I guess that's the natural language perspective. And for BioLink, I think it's designed to be as expressive as currently the translator project requires. And so, I showed you the one example of attaching frequency to a disease phenotype. It doesn't restrict what you add. It's just, but what it contains is an attempt to standardize. So you're able to add additional pieces there, and as you need to increase that expressivity. But then, of course, you're moving away from the standardized aspect, right? And so maybe there actually is this tension between standardization globally and this expressiveness that really you just mentioned, right? And so I think that this is going to be an active area. Certain areas of knowledge are maybe easier to represent. I think precision medicine is probably still bleeding edge. Maybe we don't know those phases that well, or maybe in addition to estrogen receptor, there are other aspects of tamoxifen success, right? And so maybe there's also danger here. Maybe you're kind of alluding to that if you model something in a specific way, then it looks like that's the truth, whereas there's maybe more nuance there, right? So that there probably is a danger there. And I guess, just say you have to stay on your toes and keep yourself honest and keep asking the questions, I think.
Yeah, and the second side of my question is how much bias that we don't know about using the microbial knowledge graph in inferring new links and new knowledge.
Mm-hmm.
The reason I'm saying microbial is in this field, you know better than anyone else that all of the analysis are automated, even from the annotation, gene finding, and gene annotation is by comparing the sequence. Very few people actually do experiments like in the other field and construct the knowledge graph from multiple sources.
Mm-hmm.
So if you actually construct the knowledge graph automatically and also use this to learn, are you mostly likely to review the obvious part of the knowledge? And for very subtle one that require functional perturbation, like a gene mutation will cause completely novel way of synthesizing a metabolite, you will never be able to predict. Are you concerned with that?
Well, so yeah, sure. So I guess I would say that we currently don't include that experimental data in the knowledge graph, things like taxa or function abundance. There we have some ideas about that. So currently that trait table is a compilation from literature, right? And from textbooks. So a lot of those are old biochemistry experiments, going back even 100 years, I guess, where people didn't have sequencing and didn't have computers yet. So they had to do experiments like us. So I think there is some portion of data, and a lot of that is still locked in literature. So that's why we think these NLP approaches will be able to access. And actually, the National Library of Medicine even has a corpus of papers from that were scanned, pre-digital era. I think they go back 200 years. So there's a lot of history here, right, on microbial knowledge, and again, that's tied to microscopy in our case. So those are observations. Maybe those are kind of not fine resolution enough for a lot of these molecular questions, like you said, changing, gaining a mutation to change some metabolite production, right? That may be out of scope at the moment. We are thinking about how to approach that. I'm actually working on an internal proposal at LBL due next week with some other people that work on metagenomes to work on statistical summaries from the metagenome data, and also ways to renormalize that data, because that is actually a huge problem. I alluded to that a little bit in the sample metadata, but like you said, there are all these automated pipelines. They're working on with reference data with different versions, slightly different parameters. It is that part I think is a mess. We actually work with the EBI magnifier resource. So they have reprocessed a large collection of metagenomes and provide the abundance for taxa and functions. And so that's kind of one global data set that you can work with. It's still going to have some issues, but at least it's processed in the same way, right? But I think in order to get to maybe these more interesting molecular-based predictions, right, it's going to involve some work and also some new data sources, I think.
Yeah, I know that we're over time, but I want to give Diane questions some attention. So can you quickly address that in the chat box?
Okay, so I'm going to read Diane's question. So number one, how do we extract meaningful knowledge graph for link prediction? Is it using Sparkle queries on association edge types? Okay, so let me think about that. So I guess in our purpose, the link prediction is really a target for the knowledge graph construction and maybe other related applications. And so you include the data that you think will be informative for this link prediction, and then we'll also produce a graph that's amenable for walking, right? So one aspect they didn't talk about too much is that you can easily create a knowledge graph with these disconnected components, right, or weakly connected components, and that will introduce biases in your random walking and then in the embeddings, and probably it just won't work, right? So that's why we have to use these qualitative diagnostics to make sure that the graph is well connected, that we're seeing some relationships that we expect, and that's still, I think there's more work needed there to quantify that. Our collaborators in Italy actually have some methods that are maybe going to help with this, but it's not quite ready yet. And in terms of a Sparkle query, I see maybe you're asking about the positive and negative training data. So I think, yes, that can be definitely performed with a query to say, give me all the edges that correspond to my entities of interest, right? So that would be your positive data. And similarly, you could also create the negative ones with a query. It's actually an interesting idea. Dedicated tools for ETL. Well, yeah, that part I think is not quite scalable yet. Just because we can reuse some of the code for ETL if you're working on the same data source. So that part is easy, but there are a lot of data sources and a lot of ways to parse and interpret them, right? So I think that maybe still needs more thought and work. Obviously, if you could cast existing data as something like a BioLink standardized data set, right, that would make that process easier. Maybe it would also help you make a survey of the data out there to know what's there and what's useful. So I think that those are definitely exciting directions, and I think we would be interested in collaborating on that. So it sounds like you're thinking about the right problems. Okay, and number 2 is the link prediction application and extracted KG available for testing? Yes, so the KG you can download from KG Microbe and KG Hub. The positive negative training data, that's a Jupyter notebook. And the link predictions similarly. So that's something I could share with you. That might be in the repo. I'd have to check if that version is current enough. But yeah, if you're interested, I can certainly share that data with you.
All right. Well, thank you so much, Marcin. That was a fantastic talk, and adds the layers of knowledge that was not very frequently used at UAB.
Okay, interesting. Well, thanks for having me. It was a pleasure, and it had really great questions. I think discussion is very useful for me.
Okay. Well, on behalf of all the audience, and let me just give you a round of applause. Thank you so much. All right.
Thank you.
Okay. Take care, everybody. Bye.